with anti-diabetic therapy. However, I also was participating a lot when our institution just started creating and designing microbiome studies. Therefore, I learned a lot during the process. And today I will share our experience and other recent information in the literature about creating your own microbiome studies. So shortly, what will I tell more within today's lecture? So we will talk about questions that we need to answer before we start or sampling. Also, we will talk about the main study designs and some specificities on them, and also power calculations, which is a quite important question in every type of study. Then we will shortly look over the most popular methods, benefits, and some shortcomings of them. We will shortly talk about sampling logistics, sample type, sample transportation and storage, and also from the further processing, we will shortly touch DNA extraction and also talk about contamination, which is really important to gain trustful data and results. So we know that microbiome can be found in various locations on both human body and also the environment. I will not repeat the microbiome definition this time, assuming that you have already idea about that. So the thing that I want to emphasize in this slide is that most of the things that I will tell today will be as example of human microbiome studies. However, they apply to various types of microbiome populations and can be used also in environmental studies if needed. So this slide just shortly shows that from the creating a study and designing a study and to obtaining data in your computer, there are a lot of steps to be taken. So we will talk shortly about them, and each of these steps should be thought out. And there are numerous things that we should take into account before we proceed, because we would like to get these results in high quality and to be sure that everything before this is done well. So we will start about some questions before sampling. So firstly, we would need to see what is the aim of the study. And there are various approaches that can be used. We need to see the target. We wouldn't want to realize that something is missing when the samples are collected, the intervention ended, and therefore we need to ask these questions, and I think this slide could help us. So there are, of course, many other approaches, but if we try to categorize them, then we could see that here are four main categories of the analysis approach that can be used in microbiome studies. So firstly, if we just want to know who is there and what they can possibly do, then it's most likely some DNA-based approaches. So it could be both targeted analysis of microbial DNA and as well shotgun metagenomics. If we are more interested on how do they respond and what pathways are activated, then we would be more interested in metatranscriptomics, and we will talk about it later. If you want to analyze the RNA, there are other precautions that should be taken into account already at the sample collection step. We just can't, after collection, think, oh, I want to see the RNA as well. Maybe the samples are not suitable for that anymore. And of course, we would like to know how are microbiome interacting with the host and what proteins are being produced there, and maybe also what chemical outcomes of their activity can we detect in the microbiome. So therefore, we would use protein-based approaches or metabolite-based approaches. And so again, we just should ask these questions for ourselves to find the aim. And when we know what we want to look further, we can prepare for this during the study design and also during the sample collection. So let's talk about study design. So in general, when we are creating the study design of microbiome studies, what we need to remember is that microbiome is characterized by high variability. So we know it's individual in each person. We know it has high variability across various body sites, across geographical localizations, and so on and so on. So there are various factors which make the study design a bit more tricky than maybe in sometimes when we analyze some other biological samples. Usually when we create study design and we create microbiome studies, we should define specific inclusion-exclusion criteria. Why it's important? Because therefore we will create more homogeneous group, and we will also increase study power. So there could be, we could exclude some patients who have been using antibiotics for within the past 2 months, for example, or we could narrow down the characteristics of the disease that we are looking or analyzing. So these criteria should be defined as, so that's some way that in which we can reduce this variability in our study group. We also need to define healthy controls if needed. That could depend if you are using longitudinal or case control design. And you see that healthy controls are marked there because it's quite difficult to define what is healthy and what is healthy microbiome and so on. However, usually according to the literature, if we want to define healthy controls as closely as possible, then usually it says that they are people in age range from 18 to 64, and they have no chronic autoimmune, gastrointestinal, or oncological diseases. Of course, this could differ if you are, for example, studying some of the diseases. But per in healthy controls, you would like to avoid them because it is known that they can heavily impact the microbiome composition. And of course, you can add some additional diagnoses that you would like to exclude, depending on the study type that you are performing. Then, of course, we know that we can't control everything. We can try to control a lot of things, but especially if we are talking about human studies, the next best thing that we could do is collect metadata. So additional information about factors that could impact the results. Therefore, when we see the results, maybe there's some unexpected clustering or some outliers or some other interesting results, we can go back to our additional data and see, well, there's the explanation. This person has used antibiotics, or this person has some specific health condition that we thought that wouldn't affect the results, but we see that it is affecting. So therefore, if we can't control some factors, we can collect information about that. So as I already mentioned, we could collect information about antibiotic and other medication treatment, collect information about other diseases, diet, which could also impact our results, as you know, especially if we talk about gut microbiome. And of course, there could be other factors that are specific to your study. I like this example to show that, for example, if you're studying skin microbiome, it's said that you need approximately 2 hours to rebalance the microbiome after washing hands. So you should take that into account and so on. So if you're working with environmental samples or some other microbiome subpopulations, there could be some factors that I can't imagine at the moment, but I'm sure that they could, they are already shown somewhere in literature, or you have found them yourselves. So let's talk in more detail about some specific study designs. So the first one is case control, which is quite often used. To perform these studies with highest quality, it should be good if we could match the study groups. We could match them by age, gender, BMI. As we know, these factors can also impact the microbiome. We need to take into account that nevertheless, however we try to match the study groups, there will still be high intra and inter-individual variety because of the differences in lifestyle, diet, medication use, physiology, geographic location, and so on. So maybe some of these factors we can control, and for some others, we just need to collect the information like we talked in the previous slide. And I actually really like this suggestion that if it's possible, maybe we can create more than one control group. So we have used the case control, so healthy disease, using medication, doesn't use the medication. But so yeah, we could use like this healthy group. And maybe if we are looking at some specific disease, we can also look at compare patients with specific symptoms and asymptomatic patients. So that could also be some interesting control. And maybe we could compare some different stages of disease or the therapy response or whatever we are looking at. So I think this is also a valuable and practical suggestion, of course, if it's possible to implement, taking into account financial and other aspects of the study. And also time of sample collection can be crucial because there has been some studies showing that microbiome can be affected by circadian rhythm, so time during the day. And moreover, if we are talking about seasonal effect in countries where it's more pronounced, then that could affect your results significantly. For example, in Latvia, during summer, we would eat a lot of strawberries. I don't know, during June, a lot of cheese and beer. And if you look at December, then we know it's time for some citrus fruits, gingerbread, and other Christmassy foods. And if we would, like, for example, collect cases in the summer and controls in winter, we would see just some differences because of the seasonal effect during the sample collection. So that should be taken into account as well. So I don't know, the best could be collect both. So we know that we, for example, we need whole year to collect samples, then we should collect both some controls in some cases in June and some controls in some cases also later. So so the effect of the seasonal effect is not so pronounced and has not, doesn't impact your results so much. So, and just as a fun example, if we would want to create a perfect control group, then science is saying that that could be monozygotic twins, as you can see here. So it has been shown that the microbiome is more similar between monozygotic twins than dizygotic twins. Of course, we need to take into account that if they start to living apart, these differences start to increase. However, there have been many studies employing this factor and analyzing not only genetic influence of the host on the microbiome, but also some specific health conditions, just trying to employ this almost perfect control group. So next, we will also shortly talk about longitudinal design. So there are many questions within longitudinal design. So how many time points we would like to analyze? Would it just be before and after, or would it be time series? That would definitely afterwards should be taken into account when we perform statistical analysis, because some tests can be used for one and can't for the other. Also, if the longitudinal study will be observational or will we intervene with some medication treatment or dietary changes and so on. And again, of course, seasonal impact sample collection time if we are talking about for example, 3 months long longitudinal study, then the seasonal impact wouldn't be so significant. But if we are talking about follow-up during a year or even more, then that should also be taken into account. Of course, there are many benefits for a longitudinal study. Mostly, the first sample in longitudinal study can be used as a control, which is really important benefit, especially if you could, if it's possible to collect the sample before the intervention or before the observation and so on. For example, we collect samples from newly diagnosed type 2 diabetes patients, and we get the first sample before they start any treatment. So that is a significant control. And it also has been shown that if you use a longitudinal study, then we get increased study power. So we have reduced variability because each individual is kind of control for themselves, and therefore the study power is increased. Of course, if we talk about challenges in performing longitudinal design, then I think I would say that the most significant one would be adherence to the study, that we should take into account that, especially if the time frame is quite long, some patients may start antibiotic treatment and would so they would not be suitable for your study. Some just wouldn't want to continue to participate and so on. So there would definitely, you have to take into account the possibility of dropouts from your study group. And of course, short example also of longitudinal design. I really like this study. It's quite recent. So they analyzed microbiome dynamics in inflammatory bowel disease, and they tried to define a healthy plane. So looking at dynamics, it means that they used longitudinal design. So they plotted all samples in this multidimensional axis. And so they tried to define a healthy plane, which is here with the gray color. So it is a surface where most of the healthy samples could be found. And what is interesting, that some IBD patients periodically were on the healthy plane. So what would it mean if we would use case control design? In some cases, we could not detect the differences between the healthy controls and the IBD patients if they were on the healthy plane. However, when we use this longitudinal design, we can see these fluctuations. And we can see that, which is also important, that the IBD patient microbiome is fluctuating more in general, that the healthy microbiome is mostly staying in the same localization, you could say, if you depict it in this way using the beta diversity. So this clearly shows some interesting advantages of the longitudinal design and things that you couldn't see if you just used case control design. And ending the first part of this lecture, we will also shortly talk about power calculations. And I assume that a lot of you know that, of course, power calculations are important. They are widely used within most of the biological studies and so on. However, what is challenging that in microbiome analysis, we still have more questions than answers when we talk about study power calculations. There are some methods that use the following algorithms to calculate study power. However, it is still quite tricky. There are scientists who are sure that study power should be calculated and try to implement them, but a lot of scientists still believe that there are more challenges than answers with this, especially if you are talking about more complicated designs like longitudinal design. I myself have read a lot of publications saying we should calculate power in the microbiome studies, especially for longitudinal studies. And that's it. they maybe have some suggestions, but they don't have the answers. They just say that we should do it. However, it's all not so bad. There are some tools available for case control studies. For example, this one, it is created on R Shiny, where you can just, it's quite user friendly. You can just change some parameters and obtain information about the expected study power. However, as I said, for longitudinal studies, I haven't seen such tool. Hopefully one day it will be there. But other way how people are performing these studies and trying to still take into account study power is afterwards, when they collect the samples, they calculate the power on specific parameters. So after that, of course, that is not the best approach, but currently, as we see, we do not have a lot of alternatives in the microbiome study field. And in conclusion, in the first part, so for designing a microbiome study, we talked about lots of things, and I will leave in my slides here. Here you can also see a publication from which it has been taken, a quite nice checklist about questions that you should answer when you create and design your study. And this is not only about sample collection, but also about bioinformatic analysis and other steps. So maybe this could be useful for you in practice. So next, we will talk about methods. I will try to present a short overview of the most used approaches for microbiome analysis, and also to give information about their benefits and about their shortcomings. However, I'm not proposing that today I will tell you everything. That's definitely not possible. So I will talk about some most often used approaches, and of course there could be some additional benefits or shortcomings that I'm not mentioning in this lecture. However, I will try to highlight those that are most important. But yeah, there could be additional ones. So this is, I think, quite often seen picture. So we have our sample, and we can do, for example, culturomics. We can grow the bacteria and analyze them in that way. We can do the genomic analysis, the functional analysis. So this is quite similar to what we already talked in one of the first slides. And I will shortly, as I said, go through the main methods that are being used in this. So we will start with culture dependent methods. The next few slides will be organized like this. Like I will have the pros, I will have the cons of the method, and here will be short information about what we can analyze, what microorganisms from the microbiome, and what is the amount of data most often obtained. So if we talk about the positive sides of culture-dependent methods, it is quite a traditional approach for characterizing bacteria, as well as some archaea and viruses. It allows to perform functional in vitro experiments. They are really significant for a lot of further steps. For example, they are basis for development of probiotics. You need to be able to culture them. And of course, you can, in the in vitro experiments, you can add some other metabolites or some medications that you're studying, and to evaluate the reaction of some specific microorganisms in vitro environment. And it's also possible to combine this method with other, for example, with DNA and RNA-based methods. So we can try to grow these bacteria or other microorganisms using this method, and afterwards we can sample them and extract the DNA or RNA and sequence and use some of the methods that we will talk more about in the next slide. If we talk about shortcomings of this method, of course it's complicated, mostly due to anaerobic microbiota. If we talk especially about human gut microbiome, we know that a lot of microbiome there is anaerobic. And so we need to have specific equipment. If we want, for example, even to culture some already collected samples, they need to be collected in a specific way, and so on. And of course, there is interaction between taxa. For example, I have seen experiments where the bacteria are grown in layers using filters between them. So you can get a pure culture on the top layer, but on the bottom layer, there are some other bacteria growing, and the filter is allowing some metabolites to go through. So some bacteria, they need interaction with other microorganisms so they can grow. So it also makes it quite complicated to find out which other microorganisms are needed for the growth of the bacteria or other microorganisms that you want to analyze. And of course, the information is limited to the corresponding knowledge and skills. We still, if I think the latest that I have read, that still approximately half of the human gut microbiome, we do not know how to culture in pure cultures. So it's still challenging. It's getting better. Of course, some years ago, it was more than 70% that we couldn't grow. But so yeah, sometimes we just don't have the information how to do it. So it is limited. And so this is, yeah. Saying the similar what I already said. So it's relatively low percentage of the microorganisms that we know how we can culture. Next method would be PCR-based arrays. So the benefits of it that we could analyze one or some number of specific microorganisms that we want. It's really fast. It's suitable for diagnostics and forensics. We can see here some examples. If you just want to know if some pathogen is there, it's fast, and also I would say cost-effective method for obtaining the data. However, data are limited to the array, so we wouldn't see any information outside that, and there could also be some PCR artifacts within our results. Next is analysis of marker gene amplicons. I would say that this is currently still be the most used method for human microbiome and other, I think also other microbiome analysis. So we have various types of, I could say, barcodes that can be used for this. So we have 16S rRNA for bacteria and some archaea. We have 18S rRNA for eukaryotes and ETS for fungi. So we can choose what we want to see, and it's also quite fast, and yeah, it's widely used. I think maybe nowadays due to the cost effectiveness, metagenomic sequencing may catch up to this quite soon, but I would say, yeah, the most data are definitely in these marker gene amplicon methods. We can obtain information about most of the present microorganisms, of course, limited on corresponding to the marker gene. However, if you're interested in bacteria, that could be enough to answer our questions. Also, there is low biomass requirement, which is really important for some, I would say, maybe more exotic microbiome locations. Also, for example, we know that skin microbiome has a lot of host DNA, therefore this 16S analysis is really suitable for that. For example, also blood microbiome or other specific microbiomes where the biomass is really low, this method is really good. Also, it's suitable for samples with high abundance of host DNA, as I already mentioned, the skin microbiome, the oral microbiome, and so on. And there's high number of publicly available data, which could help if you want to compare your results with other studies. So that could be also an important factor. If we talk about shortcomings of this method, the number of marker gene copies can be variable. Of course, we would like to expect that one bacteria or other organism has one copy of marker gene. However, it is not the true. There are some species that have multiple copies. There can be that could definitely impact the results and create some deviations from the true. These methods mostly can classify up to genus level, and that could not be enough if you're interested into species or strains. And resolution depends on the chosen marker gene and its region. As we know, for 16S RNA, there are 9 variable regions, and they have been widely characterized. That, for example, I think V3, V4 are quite often used, V6 and so on. But for example, V8, V9 are quite rarely because they can't distinguish a lot of the microorganisms in the samples. And of course, we have no information if the microorganisms that we detected are alive, dead, or active. And of course, there also could be some PCR artifacts and primer specificity. We know that we design primers mostly for the conservative regions of these genes. However, there could be some circumstances where the conservative regions are not so conservative, and the primers are not specific to some species, and therefore we can't obtain data about them. And of course, we could get some false positive results in low biomass samples. That could be just due to the fact that we know that microbiome is everywhere. Therefore, we could have some contamination from the environment. And of course, we know that the reagents for PCR are also obtained from microorganisms. So there could be something left in the reagents, and we could get some false positive results. And of course, we have limited data about functions. Using this method, we know what is living there, and we could just, for example, search literature about information what they could do, but we can't be sure about any activity. There are some prediction tools that try to predict functions from 16S RNA data. However, they're not quite often used, and I think the fact that the shotgun metagenomic analysis is getting cheaper and you can get some more detailed information about functions in there, I think people most often then go to this more advanced method. So we will also talk shortly about shotgun metagenomics. So this method allows to obtain us most of the available genetic information in the sample. Of course, it can be dependent on the sequencing depth, and resolution is up to species and strain level, which is really, really good. We can also get information not only about taxonomic profile, but also about functional profile. So it would depend on the database or analysis tool that you are using in the bioinformatic processing steps. But for example, with human tool, you can get gene families and pathways. So you can't be sure if they are active, but you can see the potential of these microorganisms, what they could do. And we also can perform de novo assembly for microorganisms that are not in the databases that are currently available. If we talk about the shortcomings, then of course part of the data is taken by host DNA, because we are analyzing all the DNA that is in sample. In some samples it's okay. For example, in fecal microbiome, it's okay. The host DNA is not in such a great amount that it would significantly impact the obtained results about microbiome. And of course, we use bioinformatic tools to filter them out. However, if we would want to do shotgun metagenomics on mouse microbiome or skin microbiome, we would get 90–95% or even higher amount would be human reads. And of course, there are also labs that are performing this, like extra deep sequencing, also for these kind of microbiome subpopulations, but of course it's definitely more expensive. There is still incomplete information in the reference databases. There could be some mistakes there and so on. And if we want to do a comprehensive microbiome characterization, high sequencing depth is needed. So that again that needs to be taken into account, can impact the expenses of your analysis and so on. And we still don't have information about whether the microorganisms are dead, alive, or active. And if we use the de novo assembly, of course, we can have some artifacts there as well. Next, if we go more into the functional analysis field, so if we would like to use the Metatranscriptomics method, this method can identify live microorganisms, but it should be used in combination with marker gene analysis. And it gives information about the dynamic interaction interaction and dynamic differences between individuals and so on. And also it gives information about the microbial activity, including the response reactions to the intervention that we are performing and so on. It is quite complex method if we talk especially about sample collection and storage, because we know that RNA is more stable than DNA. Therefore, starting from sample collection, we whether need to use some specific preservatives or temperatures and so on, so we don't lose and degrade the RNA that is in samples. It is quite expensive and complex in sample processing, as with other RNA-based methods, but it's getting, as we know, the sequencing is getting more cost-effective, the prices are lower, therefore this method is also getting a bit more affordable. We have contamination from host mRNA and rRNA, because we again are sequencing all the RNA that is in the sample. And data can be affected by microorganisms with high transcriptional activity. So they can be like outliers, and you would see that you have changes in some transcripts. However, it could be just one or two specific species creating that. And as I mentioned here, you can detect microorganisms, but you need to combine it with DNA sequencing methods to distinguish transcription levels from specific changes in abundance of taxonomic units. So if you really see that the transcription is increasing, or just the number of the abundance of the species is increasing, so on. So this is, again, increases the expenses. However, the information that you obtain is, of course, next level and really interesting. And in the end, about metabolomics and metaproteomics, you can use a targeted method for detection of known metabolites or proteins like bile acids, some amino acids, and so on. And you can use untargeted methods for search of new compounds. It gives opportunity to evaluate also functional changes. So next level after the transcriptional changes. However, it cannot, most often cannot be connected to specific microorganisms. So we see increase in some metabolite, but we cannot say which bacteria or other microorganism is producing it. It's quite expensive and complex. And there are differences in stability of study subjects. Some metabolites can, for example, be stable if frozen for a week, some for months, and so on. So you need to take that into account when you're analyzing your data using these approaches. Of course, there are other methods I will just shortly highlight. For example, this is called MapSec, where it's like spatial characterization of microbiome. if you want to know more, you can just read this publication. I just found this is quite interesting example of a new approach how to analyze microbiome. And of course, also single cell analysis is having increased popularity also in microbiome studies. So that was shortly about the most used methods. I hope that I highlighted the important things for you. And now we will shortly talk about sampling of your microbiome samples and the factors that most significantly could impact the data. So let's start with logistics. So again, there will be some questions that you should answer or think about when you collect your samples. For example, who will collect the sample, whether it will be a patient at home or whether it will be a medical professional who will do that. You should ensure precise instructions for collection independently, whether it's the patient itself or the medical professional who collects the samples, as we know some mishaps in sample collection could impact your results significantly. We also need to take into account patient safety, especially if we are using some collection tubes with preservatives. Are they harmful? Do we need to provide patients with gloves? Do we need to add some specific instructions? Do not eat that, or some other things, because imagination of people is really interesting and surprising sometimes. So you need to take all possible precautions. You can also ensure patients with some assisting devices like this one you can put around the toilet seat that helps to collect the sample. And also you need to think about transportation, whether it will be, how soon can you transport the samples from the collection time, whether it will be possible to transport them frozen or not, because you wouldn't want to freeze and thaw the samples and so on. And of course, I think there could be another lecture about some other logistic aspects in animal studies, but in today's lecture, I won't go into deep about that, but you should, of course, there would be some specificity for that. Then, of course, we could have various sample types that we are collecting. For example, we could have just frozen sample without any additives. We can have sample with some specific buffer for sample stabilization. Also some swabs. Also during the biopsy, there could be, we could get tissue samples, we could get washout from the lumen and so on. And of course, we could have environmental samples, and each of these samples they will have some differences in the collection protocols, in the storage protocols and so on that you need to take into account. Shortly about sample transportation and storage. So the best method would be immediate freezing after the collection, but of course I know it's not always possible, especially if you're talking about patients and sample collection at home and so on. There has been some studies that questionably and are discussing some long-term, and there's question how long is long-term storage at +4 degrees. However, they have shown that there's some fungi overgrowth, and many studies have fortunately showed that it is okay in most cases to store samples, especially fecal samples, in room temperature up to 24 hours, that there are not significant changes in the microbiome composition before that and after 24 hours. Of course, if it's longer, then there start to be some fluctuations which are not good. As I already mentioned, you need to avoid thawing freezing cycles, as it impacts the microbial composition. It degrades the DNA, RNA, and so on. And of course, you can use some stabilizers. There are some examples, RNA-later and so on. But what you should take into account if you are using the stabilizers, you should check out what methods can you perform afterwards. For example, as I know, RNA-later is not so compatible with metabolite analysis afterwards. And yeah, you should take into account again what you are going to do after you collect the samples before you choose these steps. And of course, I just highlighted some of the most important aspects of the collection and transportation and logistics, but there are wide amount of publications already available. I would suggest that if you have some specific questions about storage conditions and the preservatives to be used, you can definitely look into PubMed and find that I would say most of them already are analyzed and compared, and I think you will find your answers, some specific answers, just looking more in detail on the already existing publications. And of course, if you are not sure, you can do small benchmarking studies before doing the large study, like for example, collect some experimental samples yourselves and evaluate some storage conditions and so on. So next, shortly, I will also talk about DNA extraction. Of course, there are various kits for extraction available, and I will not go into detail about these reagent complex that everyone can choose freely. There's mostly, like, it could be financial aspect, it could be dependent on the where you are localized, what kits are available to you and so on. However, the first step in the extraction of microbial DNA, and I would say also RNA, is really important because you have to make a choice whether you are using enzymatic or mechanistic cell degradation. So what it means, it's quite straightforward. Enzymatic degradation would be some reagent, and enzyme-based degradation of cell walls, and mechanistic usually uses some beads and physical force to perform the cell wall degradation. Why it is important? Because as we know, bacteria can be classified in gram-positive bacteria and gram-negative bacteria, and for the gram-negative, the cell wall is contains thick peptidoglycan layer, and if we use too mild method, we could get situation where we don't obtain information about all gram-positive method about all gram-positive bacteria, and if we use too harsh method, then we could end in situation where we degrade not only the cell wall of gram-negative bacteria, but also the DNA or other genetic material that we will want to analyze. And mostly mechanistic method is used, and it would be a golden standard. And actually, I think more often, I have written here in some cases, but I would say that nowadays, more often also a combination of these methods, that they have some beads, like you can see in this picture, and they also have some enzymatic reagents added to the sample that helps to degrade the cell wall. And you can see here that various parameters are compared, and each color of the box plot represents different extraction kits. So you could see that there are definitely differences in the parameters and also the bacterial composition. But I would say that it is expected, and the most important thing would be to, if you start your study with one extraction kit, you should stay with that extraction kit throughout all the study, and then therefore ensuring that the results are homogeneous. I will try to shortly, as I see my time of the lecture is ending, I will try shortly also highlight effects of sample processing on the study results. So here what we see combination of extraction kit, preservation buffer, and time. So each column is a different extraction kit. So you can see here the name. Then we have first two rows are samples analyzed just after collection, and the last two rows are samples analyzed after 14 days of storage, I think in room temperature. And there we have whether it's just collected frozen or it's just in some specific preservative. And what we see that actually, if we look at these pie charts, I wouldn't say that I would recognize that all these pie charts represent exactly the same sample. And I would think that you would agree, for example, here we have quite high abundance of this red bacteria. However, here it's quite small amount and so on. So we see that all these factors significantly impact the results. However, the good news are that subject specificity can be observed independently of the workflow. So here we see each individual, and if we compare here, so each column is different workflow or kit or so on used, and we see there's variation. However, I think we could still distinguish, for example, the D2 patient from D3, so there's still individual specificity is still higher. And again, of course, I understand this seems scary. The samples, the one sample could be so different depending on the results, on the reagents and workflow used, and of course, it highlights the difficulty to compare our data with others that have already been performed, with other studies that have been published. However, the subject specificity is high, and again, I would like to emphasize that if we stay with one workflow during the whole study, it should be okay, and we should be able to trust our results. And in the end, shortly about DNA processing. So we saw this picture. We already talked about a number of important aspects in the key steps of obtaining microbiome data. And in the final part, I would like to talk about another important aspect which impacts actually most of these steps. So as we know, microbiome is everywhere around us. So there is significant contamination risks that there are significant contamination risks that we should take into account and be aware and do some things to protect us from them. So about contamination risks, there could be various. We could have contaminated DNA, RNA, or other type of sample during the collection. We could have cross-contamination while we are working with the samples in the lab. We could have some environmental contamination and contamination in the reagents. I already mentioned that most of the reagents we obtain from microorganisms. And of course, we are happy that we have increased sensitivity of these massive parallel sequencing techniques. However, it means that also we have increased contamination detection, and that could increase the or confound the interpretation of results. And there are also some discussions that maybe these are some of the reasons why we obtain controversial results within other studies. And I think this picture quite significantly shows the impact of contamination. So we see that the original sample is just, like I would say, 2 blue bacteria, 1 yellow. So this is the same in both samples. And in this side, we add some red bacteria. In this side, we add a bunch of other bacteria, which is our contaminant DNA. And if we look at the results, we see that these graphs are definitely different. And if we look at the PCI, the samples are clustering totally opposite sides. And if we go a step back and remember that these are actually identical samples, just the contamination is what differs. So that, of course, makes us worry and makes us admit the significance of the contamination in the microbiome samples. And what we can do, we can sequence some controls, for example, positive and negative controls, and so on. And this also is quite interesting picture showing that if we work in plate here, like 96 sample plate. The red spot is where we put some specific bacteria, and this is, I think, yeah, if you work using automation, how far the specific bacteria have gone. So it was inserted only in one spot, in one well, but it has gone like quite far. You can see in various places of the plate. However, if we use manual work, then this contamination is actually quite, I would say, significantly smaller. But yeah, this need to be taken into account, and there's a short example as well that this is especially important if we talk about low biomass samples, because here you would see that the black is one specific bacteria, and each column is dilution of this sample. And as we dilute the sample more, we see that the actual bacteria after the processing is reducing, and we can actually, when we dilute the sample, it's like 5 times, there's 90% of the results are contamination. So this seems quite scary, but it's true. And in low biomass samples, it's more challenging. Therefore, the controls should be implemented. However, the thing is that I would say we cannot avoid this contamination risk, but again, we can prepare, and we can collect these positive and negative controls and other samples. We can collect environmental samples, for example, as some control. And of course, for example, for gut microbiome, it would be quite the contamination wouldn't be so big, because we know that most of the bacteria that can be found in our gut microbiome cannot be found on the table and so on. However, if we are analyzing, for example, skin microbiome, the situation definitely will be more tricky. And in the end, I will leave you with this small chart that also could help you. So there is, I won't go into detail, but in every step that you are doing when performing your microbiome study, there are a lot of things that you can do to prevent or to control the contamination. Also in data analysis, there are amazing tools created that could help to filter your data based on the result you obtain in the negative controls. So I would say take a more deeper look into this. There's also, yeah, you can find the publication, and I hope that this was useful, and this will be useful for you also in your future studies that you will perform. And with that, I would like to thank you all for listening and for being here. I hope it was useful and made you think, and will help you to ask these questions before creating your study, or even improve already ongoing studies. And yeah, I need to thank the Professor Jānis Kloviņš group, from which I'm a part, a postdoc of this group, because it group allowed the possibility to learn all this knowledge about microbiome studies. And yeah, that is all. I think that I talked for a whole hour. So thank you for listening. And I don't know if you have some questions, maybe there is some short time that I can answer, but if not, yeah.
Yeah, you can think about your questions. I have a